welcome you all to, as I mentioned, our first IIDR rounds of the new term, and we're going to hear today from Doctors Mark Loeb and Matt Miller about learning from COVID-19 to prepare for future pandemics. I just wanted to make sure I read that off correctly. So Mark and Matt are no strangers to those of us in the IIDR. Mark Loeb is a professor in the departments of Pathology Molecular Medicine, Health Research Methods, Evidence, and Impact at McMaster. He's the co-director of the McMaster WHO Collaborating Center for Infectious Disease, Research Methods and Recommendations. He holds a Canada Research Chair in Infectious Diseases, and is absolute rock star, as we all know. Mark's research focuses on vaccine clinical trials, and today he's going to talk about correlates of protection against COVID-19 infection and severity of disease in unvaccinated Hutterite communities, a really unique and exciting project. Our next speaker after that is of course, the director of the IIDR, Matthew Miller. He's also co-director of a new entity called the Canadian Pandemic Preparedness Hub, and an associate professor of biochemistry and biomedical sciences here at MAC. He holds a Canada Research Chair in Viral Pandemics, and his research focuses on research into vaccinology for influenza, as we all know, and of course now we're all involved in COVID-19 mitigation. And today he's going to talk about this new entity, this Canadian Pandemic Preparedness Hub, that I'm really excited that IIDR is taking a leadership role in. It really is something that I think really quite positive for us. So before I let Mark get started, I am just going to remind you that we're going to have a question and answer session. Type your questions into the Q&A bar at the bottom of the Zoom window, and then I will get to them and moderate them as best as I can. So take it away, Mark.
Okay, great. Well, thank you so much. So as Jerry said, I'm going to be talking about correlates of protection against COVID-19 infection and severity of disease in an unvaccinated Canadian Hutterite community. So the background for this is that, as you guys all know, over the course of the pandemic, vaccine uptake became widespread in most communities worldwide. And there have been community studies that have attempted to correlate pre-infection immune status with the risk of subsequent infection. And it's been done in various groups, like there have been occupational group cohorts. One was in a Swiss longitudinal police cohort. In fact, there's another one that was done in a Canadian paramedic study, just following people forward in time. There have been relatively few household cohort studies that have been conducted to address this question. And one was conducted in the pre-vaccine era in Nicaragua. This was by Eva Harris and her group that we collaborated with a lot for dengue. And another in a highly vaccinated population in Israel. So most of these, though, are post-vaccine era. They're in the era of vaccines. So less is known about the correlates where in entire communities, vaccines were seldom used. And the other piece of the background is that although neutralizing antibodies have been the most commonly tested as predictors of infection, there have been relatively fewer studies that have used anti-spike IgG levels to correlate. There have been studies that have correlated neutralizing antibodies and IgG antibodies, but there's few that have, relatively few that have used IgG antibodies as correlates of protection. And also there have been relatively few studies that have used detailed surveillance data to capture confirmed RT-PCR infection prior to doing the analysis period. So in other words, there are relatively few that have used actually surveillance to confirm natural infection prior to doing an analysis. So in this study, we followed a largely unvaccinated cohort of members of the Hutterite community to estimate the association between baseline immune status, that is using IgG antibody concentrations, and the risk of subsequent infection. And we did this throughout the pandemic. So it was throughout all the waves, through the original Wuhan strain, Alpha, Delta, and Omicron variants in Canada, in the Hutterite community, from starting in November 2020 and ending in August of 2022, at least for the analysis that I'm going to describe now. But we're ongoing. We have a phase 4. And likely later this fall, we'll be going to phase 5. So phase 1 was the initial additional cohort from November 2020 to June 2021 that was funded by CIHR. Phase 2 from July 2021 to October, and that was funded by CITF. And then from December to August 2022, there was another, the phase 3, which was funded by CITF. And that's sort of Omicron circulation. And that's really what I'm going to be focusing on in terms of the analysis to date. And the studies were done in Hutterite colonies in Alberta and Saskatchewan. So these are colonies where there's a lot of mixing of individuals, there's communal dining. It's really communal living, and with each colony having roughly anywhere from about 80 to 120 members, and a lot of children. Up to a third of the colonies involve children. This is a slide just showing the distribution of the colonies for this study in Alberta and Saskatchewan. So any member of a Hutterite colony that was enrolled in a study colony, or living in a study colony rather, was eligible. So of any age, obviously they had to provide informed consent, or assent. Children were involved, and there were absolutely no exclusion criteria. So we left it very, very open. And at baseline, we got demographic information, age checks, their past medical history, medications, the vaccine history. All of that stuff was obtained at enrollment. And we used self-collected flock nasal swabs at baseline. So whether they're symptomatic or not, we started off right off the bat getting swabs. Serum was obtained for IgG antibody, and this was tested through Matt's lab. And we got antibodies at baseline, midpoint, and at the end. So I showed you all these sort of phases. So at the end of one phase, the last set of sera would be the end titer for one phase, and they'd be the baseline for the subsequent phase, as long as the phases were sort of contiguous. So in terms of the first phase, the follow-up, so this was usually in the original strain of SARS-CoV-2, we assessed for signs and symptoms of COVID-19 twice weekly. The participants were all given symptom diaries, and essentially if they had 2 or more signs or symptoms, I'm not going to read them all out here, they were asked to self-swab, and we previously validated that it works quite well. And these were tested for RT-PCR for SARS-CoV-2, and they were tested either through the Alberta Provincial Lab for specimens from Alberta, and those from Saskatchewan were tested at St. Joe's through Mark Schmeye's lab. And we hope that the MacArthur lab will be doing the sequencing. We're looking forward to that in a couple of weeks. We also had this cluster case definition that if there were 3 or more cases over 48 hours, all members of the colony, whether they were symptomatic or not, would have swabs obtained. And then this would occur daily for the first 7 days, and then twice weekly for another 4 weeks. So as you can imagine, this is a lot of swabs, tens of thousands of swabs. In terms of follow-up, things changed a little bit for phases 2 and phases 3 because, it's a bit of a mutiny. Everybody, the field staff were saying, we can't do this anymore. There's just too many swabs. So we changed our approach a little bit. There we used flock nasal swabs weekly. So systematically, whether participants were symptomatic or not, everybody got a swab. So we were able to look at both asymptomatic infection and symptomatic infection. And we also used a symptom severity score. So this is a scale that's been used by the WHO. Either there's no infection or negligible infection, mild, moderate, or severe. And essentially, negligible was up to 3 symptoms, mild was 4 to 9, moderate was at least 10 symptoms. So it was another outcome that we can use other than the binary, RT-PCR positive or negative. So I mentioned that we were using nasal swabs for RT-PCR. The SARS-CoV-2 mRNA was identified by quantitative real-time PCR, and we were using tested in Matt's lab, IgG ELISA assay against spike. And the analysis was done, really, we wanted to focus on where there was actually viral activity, where there were cases. So it didn't make sense to us to be focusing on colonies where there are absolutely no RT-PCR SARS-CoV-2 confirmed. So there had to be at least 2 cases for the colony to make its way into the analysis. We calculated geometric mean titers and 95% confidence intervals of the IgG antibodies at baseline, comparing the infected to non-infected participants, as well as doing this by disease severity based on this WHO scale that I showed you a few minutes ago. And the idea was to test the association between the baseline IgG antibodies and natural infection using a multivariable logistic regression model. And we also wanted to test the association between the same predictor variables, that is baseline IgG infection and disease severity, using an ordered logic model. So it was sort of a proportional odds ratio approach. All models were adjusted for age, sex, the existence of a comorbidity. That was just sort of yes or no. Colony size and prior infection natural infection was one of the predictor values we were looking at. And that was, we used the data from phase 2, and we actually looked at phase 1 and phase 2 together. It made no difference. So we stuck with just using phase 2 data. That's really within 6 months of having a natural infection. And the models were clustered by colony using a random effect. We also use logistic regression to assess the probability of infection given a baseline IgG antibody above each threshold. And the way I think it's the way it's set up now, it's probably not having an infection, but it comes to the same thing. So this is what the study flowchart looks like. So we started off with 956 participants in being screened, about 700 participating in our first study, with only 24 positive, so really not much action there. And then in phase 2, we screened over 1,500, ended up with 1,300, and there we were getting more positives, 169. And then phase 3, in the Omicron era, we screened over 1,500, enrolled 1,200, and we had 438 participants that were positive. So this slide shows the baseline characteristics of the 3 phases: the screen, the number of colonies, number enrolled, etc. You can see that if you look at the column that starts with 3 to 15 years of age, you see under a third, a little bit under a third are children. And the majority of participants are sort of young to sort of young middle-aged adults. Relatively few people over the age of 65, very young median age, 25 or 26. And what's striking is the number that received mRNA vaccine, hardly any, 3.6%, 2.2%, 6% in phase 2. In phase 3, there were only 0.25%. But cumulative follow-up was good. A lot of person days, close to 200,000 in phase 3. So these are the PCR results that I sort of alluded to. This is broken down by Alberta and Saskatchewan. Again, what we're focused on is, again, phase 1, not so much. Phase 2, not too bad, but phase 3, a lot of cases. So this is the epidemic curve in phase one, where, only 2 colonies were involved, and it looks like, they were probably at the end of one outbreak when we started, and then nothing. But it was sort of interesting because all the other respiratory viruses were there. So we were concerned that there was a problem with swabbing or something, or media, but that's not the case, because rhinoentero adenovirus was circulating. This is the epidemic for phase 2, and there again, more cases, bad from a public health point of view, but better for an analysis point of view. More cases both in Alberta and Saskatchewan, and a lot of other respiratory viruses, and just shows the distribution of results by colonies. So you can see overall that there were some colonies that really had no cases whatsoever. Others, there was some variability, with some having a relatively high proportion, 219. Almost virtually everybody was infected there. And then phase 3, which is the focus today. So again, a lot of variability, and if you look at, you have 1,200 people enrolled and 438 were infected. So that's a really high attack rate, with some variability from colony to colony, which is, again, it's not good from a public health point of view, but from an analysis point of view, it's optimal. And this is the epidemic curves for phase 2 and phase 3 together. I'm not going to spend a lot of time on this because it's not the point of this talk, and we're going to regroup with Matt and his group and do some analysis. This is sort of the IgG at baseline, at the endpoint after phase 1, the endpoint after phase 2, and the endpoint after phase 3. So again, one can look at fourfold rises in antibodies and look at predictor variables, and a lot of things can be done there. But you could see even at baseline at phase 1, it wasn't sort of a naive population. They had been exposed to COVID. Nevertheless, through the different phases of follow-up, there was a lot of seroconversion. So this is the analysis data set, the baseline characteristics, and these 2 columns compare those individuals who were not infected with SARS-CoV-2 to those who were infected. And you could see, the numbers in terms of age, sex are pretty similar, a similar number in the household. There were a few more, somewhat unexpectedly, that were not infected with COVID, who had comorbidities, 21% versus about 17%. This is the baseline IgG antibodies by infection outcome. So as you can see here, there's definitely a significant difference. Those who were infected had lower baseline IgG antibodies as compared to those that were not infected. And when we looked at it based on the scale of no infection, negligible, mild, or moderate, you can see some variability. There's a significant difference between no infection and negligible, not much between negligible and mild, but then it was reduced to moderate. So overall, it was sort of highly significant differences between the groups overall. And this is the logistic regression analysis for prediction of infection. So this was the univariate analysis. So you see about a 32% risk reduction with increasing IgG antibodies with a very significant p-value. No effect for age, no effect for sex. For comorbidities, there seems to be a significant protective effect actually in those individuals who had 1 or more comorbidities, reduction by 32%. Again, this is only the univariate analysis. And previous infection was the biggie, like almost a 70% risk reduction, which was really highly significant. And this is the multivariable analysis for, again, prediction of infection. This is SARS-CoV-2, yes or no, and very similar figures. So they don't really change. What happens is when you adjust for everything, the comorbidities are no longer statistically significant. Again, so the key findings is of a significant protective effect with IgG, and more importantly, with having had natural infection, which was well documented in phase 2. Then this was the ordinal regression for prediction of severity of disease. So basically, if you look at the endpoint titers, again, a sort of similar effect with IgG, and the interpretation there would be individuals who had higher IgG titers were less likely to develop more severe disease than those who had lower levels at baseline. So the effect of age and sex wasn't significant, comorbidities wasn't significant, but again, previous infection was highly significant. This is a Kaplan-Meier curve looking at baseline endpoint titers. So as you can see, we sort of broke this down to less than the 320 or less, 640 to over 10,000 and over 20,000. Again, there's sort of overlap, but at least there's a trend in what we'd expect. And this is the probability of no infection by titer. I think when we come to publish it, we'll sort of flip it around a bit. But as you can see, there is at least a trend towards no infection, probability of no infection with increasing titers. So the summary then is IgG antibodies in previous infection within the past 6 months are associated with reduced SARS-CoV-2 infection. That is Omicron, because that was the phase 3 that we were looking at, and severity of illness in an unvaccinated Canadian cohort. Now, what's important is that this is just sort of the tip of the iceberg, because there's a lot of things that we didn't look at that we're just starting to look at. And a really important thing is that what's unique about this data set is the colonies. These are entire communities. And we had planned to basically look at children and try to identify a sort of a cutoff. So here we'd be looking at an IgG titer that was highly protective in children, and then look at colonies where, let's say, some sort of threshold, 70% of the children met this titer, and then compare those to colonies where, the number of children were less than 70%, and to see if that would have a protective effect at the level of the entire colony. And when we started looking at this, we thought this is not going to work out, because there was not anything that was so strongly protective in the children. And I think given the results of natural infection, I think the way to go is to create a model where we're just looking at natural infection alone, or with a combination of IgG titers, and try to see if there's actually a herd effect that we can detect in the database. Okay, well, thank you. That was my last slide.
Great. Thank you so much, Mark. It really shows the value of your pre-existing cohorts and your ability to pivot in a crisis like this. So again, we're gonna take questions at the end of the session. So please go ahead, type in your questions, and I'll start to curate them. And the next speaker is the IIDR Director, Matt Miller, who's gonna tell us about the CP2H Hub. Take it away, Matt.
All right, thanks so much, Jerry, and thanks, Mark, for that excellent opening talk that really sets the stage for this. So it's my pleasure really to talk to you today about this new initiative called the Canadian Pandemic Preparedness Hub, which really started in earnest almost 2 years ago now. And as of yesterday, we've collectively just jumped over a huge hoop as we move forward on our way to realizing what I think is the incredible possibilities that this hub offers in terms of transforming the way that we approach infectious diseases at McMaster. So today I'm just going to give you an overview of what this hub is all about and really hone in on the strategic focus that we've developed at McMaster with a lot of the support of investigators in IIDR. So these hubs were born out of Canada's biomanufacturing and life sciences strategy, which was a $2.2 billion investment by the federal government over 7 years that came in the wake of the pandemic as a result of the recognition that Canada was really ill prepared to support the domestic biomanufacturing of anti-infectives and vaccines. The goals of the BLSS were primarily to grow a strong competitive biomanufacturing presence in the life sciences sector and to ensure that Canada is better prepared for future pandemics by increasing our domestic capacity. Of that $2.2 billion, about $1 billion was invested in primarily academic settings. And those programs are shown in the table here. So one is the Canadian Biomedical Research Fund. This was a $250 million pot of money co-administered by TIPS and SHRC with input from CFI. And that fund is really to support the research and training to enhance our biomanufacturing and pandemic preparedness pipeline. The second pot of money was the Biosciences Research Infrastructure Fund, which is a fund that is primarily administered by CFI to support the infrastructure required to realize these goals of more biomanufacturing independence. And then, of course, there was the Clinical Trials Fund, which was a $250 million investment that was managed by CIHR. And as many of you on this call will know, McMaster was very, very successful in that competition. And in fact, the Pan-Canadian consortium that was the big investment out of that fund, called Accelerating Clinical Trials, is based at McMaster with PJ Devereaux as the PI. What's really incredible is that McMaster has really become the backbone of this biomanufacturing and life sciences strategy as a result of our successes in all of these competitions. So as I noted, about 2 years ago, we started down the road to apply to become one of the biomanufacturing hubs across the country that would be responsible for administering the CBRIF and BRIF programs. And we found out in only March of this year, actually, that we were successful in being named one of these hubs. Our hub is called CP2H, the Canadian Pandemic Preparedness Hub, and it is co-administered by the University of Ottawa. And we've received $2 million to support the administration of this hub. But what I really want to talk about in the next slides is how this hub is going to support a series of programs focused on enhancing our pandemic preparedness and preparedness for future infectious disease threats going forward. Although this program also oversees the Biomanufacturing Research Infrastructure Fund, there was a stage 1 rollout of this program, again, about 2 years ago, to first support expansion and upgrading of high containment labs across Canada. We submitted an application to this program as well. That was co-led by Jerry and myself with support from many others in IIDR, including folks like Karen Mossman, Zoe Zing, and Amy Gilgrass. And this application was successful. and will support a brand new, roughly $14 million combined CL2 plus CL3 laboratory at McMaster. It will roughly quadruple the current CL3 lab's footprint and include what I think are some really important new capabilities, including the ability to house up to 3 animal species simultaneously, and containment to enable study of vector-borne pathogens. So things like flaviviruses like dengue, West Nile, Zika, alpha viruses like chikungunya. These are all pandemic threats that are becoming more pressing as a result of climate change and the increasing habitat of the mosquitoes that harbor these viruses. But it's something we're really ill prepared for in North America, because these are threats that we haven't typically considered to be very worrisome due to our climate here. And then, of course, as I noted, we were extraordinarily successful in the Clinical Trials Fund competition, with PJ Devereaux and colleagues landing this huge pan-Canadian consortium called the Accelerating Clinical Trials Program. Several other groups at McMaster also got discrete project funding in the context of this program, including our McMaster COVID-19 aerosolized vaccine group, who received about $8.3 million to advance this vaccine into phase 2 clinical trials. And these trials will be multi-centered, based out of McMaster, with supporting sites at Dalhousie and University of Ottawa. So, as noted, stage 1 of this competition basically awarded us the ability to serve as a hub and administer these massive stage 2 pots of money. So in the CBRIF program, which again is really to support research and training, the hubs are able to oversee and administer a pot of $215 million. And in the research infrastructure competition, there's a pot of $360 million to support CFI-like projects that expand our biomanufacturing capacity. So I'm gonna talk about our hub's approach to this in the upcoming slides. So as I noted, CP2H is co-led by University of Ottawa with OHRI and McMaster, of course, with Global Nexus. And the vision of our hub has been to transform research, clinical, and manufacturing pipelines so that Canada can pivot quickly to manufacture vaccines, therapeutics, and diagnostics in the case of emerging epidemics or pandemics. What's really unique about this program is that it is heavily partnered with industry, because the goal of this program, contrary to most of our academic funding apparatus, is not really to support discovery-based research, but to accelerate and promote commercialization of these solutions. And obviously, while we're great on the discovery end, we are not as effective at commercializing. And that's where these industry partners are going to play a key role. And in particular, we've really focused on Canadian small and medium-sized enterprises, because if we want to grow out the life science sector in Canada and really increase our domestic independence, helping these Canadian-based companies succeed is essential. So one of the things that's been really exciting about overseeing this program has been, I think, the transformational new approach to how industry and academia are collaborating to accomplish these goals. So our hub has benefited from the leadership of many folks at McMaster. We have an executive board that oversees the programming, and the executive board chairs are the respective VPRs at McMaster and University of Ottawa. So Karen Mossman at McMaster, As Karen is on administrative leave this year, Andy Knights, the acting VPR, has stepped in to serve as the acting chair of our executive, and Sylvain Charbonneau, who's the VPR at U Ottawa. John Bell, who many of you will know, is a former McMaster graduate and an extraordinarily accomplished scientist, especially in the area of cancer biotherapeutics, and myself are the co-scientific directors of this hub. And we have our management team, who's led by Ken Lawless at University of Ottawa and Darren Lawless in the VPR's office here at McMaster. The hub is structured in terms of pillars, and we have a really exceptional steering committee that also has a lot of McMaster representation on it, including Brian Lichty, Leila Soleimani, and Jerry Wright. So what these hubs are supposed to do is create this continuum, which I'm showing at the top of this slide, which is increasing our discovery capacity that flows into the ability to biomanufacture at GMP grade in order to support clinical testing. And obviously, this is not a one-way street. The idea is that the clinical information that we get should feed back to inform how we biomanufacture and our discovery foci. So our hub really came together because of McMaster and University of Ottawa's long-standing history in the area of biomanufacturing. Collectively, we have the two oldest, largest, and most productive GMP grade biomanufacturing facilities in academic centers in Canada. The Fitzhenry Laboratory at McMaster, which really evolved out of early research from Frank Graham and Jack Galdi on the development of adenoviral vectors to serve as vaccines and gene therapy vectors. And University of Ottawa's Biotherapeutics Manufacturing Center, which John started there and has supported a tremendous number of campaigns focused on primarily in the cancer space, but using viral vectors as cancer-based vaccines and oncolytics in many cases. We obviously want to support that with translational research and development platforms, which are supported by the Infrastructure Fund. And of course, none of this works without a strong center on training, because it's no good to have all of this capacity and grow the sector if we're not training the HQP that we need to staff those jobs in the future. As noted earlier, supporting Canadian SMEs is central to this, and all of our efforts are really focused on pandemic preparedness, although it's important to emphasize that the research platforms that we're building are extraordinarily flexible, and you'll see that these things can be rapidly adapted to all kinds of other indications, whether that be small molecule-based drugs or other sorts of biotherapeutics. So since March, we've been focused on stage 2 of this program, which is the curation of a set of proposals focused on research, talent development, and infrastructure. So just to give you an idea of the insane timelines that our investigators have been working with, the hub was officially awarded in May of this year. We had a call for expressions of interest to submit applications to these 3 areas within our hub. And that deadline was May 12th. We received 65 EOIs, and these were reviewed for compatibility with our hub's mission by our hub steering committee and executive board. Based on those EOIs, we allowed 49 notices of intent to go forward in the competition, and the deadline for those were June 8th, at which point we had an international scientific advisory committee along with the hub leadership team review these again for for quality and coherence with our hub's overall strategic direction, and ultimately arrived at a suite of 23 endorsed applications that were due to the hub yesterday, with a final submission deadline tomorrow. So as I noted, the hub is really structured into 4 platforms. Platform 1 is focused on vaccines, 2 is focused on small molecule drugs, particularly anti-infectives, Platform 3 on scalable biomanufacturing initiatives, and Platform 4 on diagnostics and clinical assays. We also have a cross-cutting social sciences platform that feeds in things like economic considerations and research on equity into all of these platforms. And of course, as noted, the overall impetus for this program was to have better cross-sector collaboration. And so we have partners from academia, industry, nonprofits, and government involved. This is a chart that illustrates the suite of proposals that we have ultimately endorsed to go forward in our hub. So you can see that they're color-coded by platform. So in Platform 1, we have a series of proposals that are really focused on vaccines, Platform 2 on anti-infectives, Platform 3 on biomanufacturing, and Platform 4 on diagnostics. And these are all supported by a complementary series of proposals that are focused on building out the infrastructure to support these research programs. I haven't noted, but should note that these programs are 4-year programs with budgets between $500,000 and $5 million per year, and research teams of about 15 individuals. So these are really fundamentally different from our typical sort of CIHR project grants. These are major programmatic team grants that are being supported. And what I'm showing you here is just the proportion of these proposals that are either led or co-led by investigators at McMaster. So in the last couple of minutes here, what I'm going to do is just provide you an overview of how all of this links together for us internally in terms of the new enabling technologies and infrastructure that we've proposed that would be primarily McMaster based. So our proposals out of McMaster include over 20 private sector partners, some of which you can see listed here. Our infrastructure proposals include the Canadian Biomanufacturing Cooperative, which is a nationwide biologics manufacturing consortium centered out of McMaster, Ottawa, and Dalhousie. The Centre for Pathogen Modification and Immune Monitoring. This proposal is led by Jonathan Bramson and really supports GCLP analysis for diagnostics, anti-infectives, and for supporting immune analysis in vaccine trials. And another initiative led by Jake Magdalene, the Collaborative Chemistry Core for Anti-Infective Drug Development, which is really focused on de-risking and advancing early drug leads to the stage of clinical trials. These collectively have an ask of over $105 million from the funding agencies. And these groups have also secured almost $9 million in support from their private partners. The research proposals that I showed you in the last slide that are McMaster based are also summarized here, with budgets amounting to about $92 million in total requests from the funding agency, with another $27 million secured from private partners. So really impressive integration, as you can see. So in the vaccines pillar, there are 2 primary McMaster-led proposals, one led by myself and the other led by Carlos Felipe and Mike Thompson in engineering. Our proposal, or the proposal that I've led, is called the Canadian Vaccine Accelerator Consortium, and it includes investigators from McMaster, University of Ottawa, and Vito. And it's focused on the development of a series of flexible Canadian vaccine platforms suitable for both respiratory mucosal and parenteral delivery that leverage our domestic biomanufacturing infrastructure. So the idea here is to characterize, de-risk, and prepare for clinical testing a series of platforms for current infectious disease threats, including things like influenza, COVID-19, and chikungunya virus, actually, but that can be rapidly reconfigured for any new disease that might emerge in the future, with the existing ability to rapidly biomanufacture these things domestically. The proposal led by Mike and Carlos is really complementary to this in that it focuses on thermostable vaccines and different routes of delivery. And all of this will be enabled by this huge Canadian biomanufacturing cooperative, which is led by Duncan Stewart at OHRI and Brian Lichty here at McMaster, that would upgrade and expand our existing GMP facilities. In the anti-infective space, we have 3 McMaster-led proposals that have been grouped basically by pathogen type. So we have an antibacterial discovery platform led by Jerry, an antifungal discovery platform led by Lindsay Callan, and an antiviral platform led by Cameron Curry. And again, all of these include collaborative teams from across Canada. These platforms will be primarily supported by Jake's collaborative chemistry core, which will facilitate massively increased capacities in the medchem and admet spaces to advance and de-risk and scale up these small molecules. And our unique area of strength is really in the natural product discovery area and in the synthetic biology platforms that have been pioneered by folks like Jerry and Eric, and now Lindsay and Cameron. There's also a proposal led by Jean Marshall at Dalhousie with a lot of input from McMaster, including and most notably Heather Sheardown in engineering, to provide new drugs that support lung healing and avoid lung injury after respiratory infection. So this sort of complements the approach of targeting the pathogen themselves, because I'm sure, as many of you know, oftentimes targeting the pathogen is not as therapeutically or clinically beneficial as we would hope. And disease-modifying drugs often have superior clinical benefits. So we wanted to ensure that we had proposals that supported both sides of that coin. On the diagnostic side, we have a really innovative proposal co-led by Leila Soleimani and Yingfu Li to develop at-home point-of-care and laboratory-based diagnostic pipelines focused on aptamer technologies. And complementing that is a wastewater surveillance platform led by Rob Delatoya at University of Ottawa and David Bellier here at McMaster to provide essentially a real-time early warning system based on automated high-throughput monitoring of wastewater. These platforms are supported by Jonathan Bramson's infrastructure platform, the Center for Pathogen Identification and Immune Monitoring. And this platform not only supports the diagnostics proposals, but also, as I noted, the anti-infective proposals and the vaccine proposals by adding in GCLP, or good clinical laboratory practice facilities, which are often required as a standard for clinical trial applications and new drug submissions. And all of this is supported by a modeling and pandemic preparedness platform led by David Earn, which will provide the data to inform all of our other platforms and essentially ensure that our hub is built for purpose. So in summary, what we're really trying to do is up our game internally on a massive scale to move from discovery through to preclinical validation and GMP manufacturing and scale up of new solutions to infectious disease threats that can be rapidly moved into the clinic and commercialized with the help of our industrial partners. So with that, I'll stop, and I would be happy to turn the floor back to Jerry to moderate the question period.
Excellent. Well, thank you so much, Matt, and let me also congratulate you and the Hub leadership team for really a Herculean effort over the last, as you noted, from the middle of May to now. There's the size of the lift on this program is almost indescribable. And kudos to you and the team, and shame on the federal government for ruining our summers. But nevertheless, we move on. So as I encourage you all to type your questions in the Q&A, we have one from Mark, from Jonathan Dushoff, and it says, didn't understand Mark's last graph, titer versus infection, was consistent with the earlier relatively strong protective effects of antibodies. I'd like to hear more about how strong the protection of antibodies is, i.e. how to interpret the protective coefficients associated with log titers. And if I was playing devil's advocate, I would say focusing on significance tells us more about how good Mark's study setup is than it tells us about COVID.
I agree about I agree with Jonathan that the focus shouldn't be on significance, and I think to me the importance is the relative effect size, right? We found a very strong effect size was infection for IgG titers. It was relatively weaker. The interpretation of the regression coefficient is just a regression is per unit increase. In the log transform titer is associated with a reduction of infection by 32%. So it's always per unit. In this case, it was per unit log transformed and titer. The point about the last graph is, it's a valid point, because we were surprised because we were going through titer by titer, and we wouldn't have expected the effect at the higher titers, right? When we looked at phase 2, it was more of a uniform increase, but for phase 3, that didn't happen. So I think we're going to have to explore that in greater detail. So valid comments.
Good. Matt, I have a question about the hub, and maybe now that we're through this phase, or we will be on Thursday. What are next steps? Like when do we anticipate? Because you can see, I think you've really explained how this could be a really comprehensive addition to the ecosystem that our researchers at the IIDR, and I'm thinking of Mark's studies, for example, how much they could benefit from this kind of infrastructure and activity happening at McMaster and training and what have you. When can we expect, answers and about the success of the funding, and when can we get a shovel in the ground in terms of infrastructure?
Yeah, thanks for bringing that up, Jerry. So these applications will progress through 2 phases of review in the coming months. First is a scientific review, and those applications that are judged to be very good or excellent in the scientific review will go on to a strategic review, which is essentially a political review for how they're judged to fit into the whole BLSS strategy overall. In February, one of the really good things, I think, actually, is that we've learned that the strategic reviewers will have a face-to-face meeting with John Bell and I to ask questions and allow us to articulate and justify the suite of applications we've endorsed and provide some narrative about how they all fit together. I think this is great because it will avoid misunderstandings and give us an opportunity to really emphasize our priorities and the cohesive way that this program was curated. Then we'll receive decisions in March, and the funding will flow as of April 1st, 2024. So we're looking at sort of, the typical 6-ish month window, which, as you alluded to earlier, is absurd because they only gave us 6 months to put this whole program together and are now allowing themselves a cozy 6 months to review. But I've been so unbelievably impressed by the resilience of our investigators who have really undertaken what I think has been the most challenging program that I've ever seen due to the complexity and the timelines to put together an absolutely unbelievable suite of applications. All of these things were reviewed by an international scientific advisory committee composed of world-leading experts in academia, industry, and government. And the feedback we got on the quality of the proposals, particularly those led by McMaster, was exceptional.
Well, that's welcome news. So we're approaching the top of the hour. I think what we'll do, there's not a lot of questions in the question section. I think we're all, in awe of both of you for the amount of work that you've done. And if I think back of, with my hat on as the ex-director of when we founded the IIDR in 2007, and if this is exactly what we were hoping for, is a quality of world-leading science and leadership, both at the experimental level and at the structural level, as that you guys have explained today, I think is outstanding. So we should all be proud of the IIDR. We should be proud of you guys, and I want to thank you for launching our rounds this term, and I want to wish everyone a fantastic term and great science to everybody. So thank you both for this, and we'll sign off now. Take care. Bye-bye.
Thanks so much, Jerry. Thanks, everyone. Bye-bye.